Assalamu alaikum. I'm Dr. Ismail. Today we will talk about another Gromacs tutorial known as membrane protein KALP15 in DPPC. In this tutorial, we will teach you how to simulate membrane protein known as KALP15, which is used as a reference protein in a lipid membrane known as DPPC using the Gromacs. The parameters used in this tutorial are budget parameters. These parameters, however, now outdated and the Gromacs website has removed these parameters. But you can find these parameters in the video description. In the video description, we have provided the links of all the files and the budget parameters file in the link. You can find the link and download all the parameters. We will use this tutorial to maintain the original tutorial of Gromacs to make available for everyone. However, if you want to change these budget parameters, you can use the modern tool like Charm GUI. We will make a separate video on the Charm GUI that how you can generate the membrane protein parameters. This is the original tutorial which we are going to demonstrate today. This is a Gromacs tutorial, membrane protein KALP15 in DPPC. So let's start the tutorial. This is the first tutorial. We will go through the each step and will make this tutorial easy for you. We will make the command list and we will provide complete command list in our video description as a link so that you can copy paste this command and run as it. We will also provide complete files in the video tutorial link as a downloadable file. To start this tutorial, you will need the Linux system Ubuntu. Kindly note that Gromacs does not work in the Windows, so you will need the Linux. We have prepared a complete video that how you can install the Linux in the Windows system, how you can enable the virtualization, and how you can install the Gromacs. You can watch this video which is available on our YouTube tutorial under the playlist of the Gromacs tutorials. Briefly, the first step is you have to enable the Windows virtualization of your system. The second step is you need to install the Ubuntu. For this purpose, you will go to Microsoft Store and install the latest version. In this window, in this tutorial, we are using Ubuntu 24. Once the Ubuntu installed, you need to run these commands one by one. The first command is to update your system. This will update and upgrade the Ubuntu system. Like if I copy this command and paste it in, it will ask you a password. This is the password you need to save when you install the Linux. So enter the password and it will start working and update and upgrade your system. It is recommended to run this command once a day. It is now completed. Then run these commands one by one. This will install the dependencies which are required for the Gromacs. Then install and compile the Gromacs. For this, you need to run these commands. We have already demonstrated in our installation demo. So here I am going a quick review. Once these are installed, you need to run these two commands. These commands will source the Gromacs from your Ubuntu. Copy this command and paste in the Ubuntu terminal and press enter, and it is done. After the installation of Gromacs, this is a small software which is known as the Grace. Simply run this command, copy this command and go to the Ubuntu terminal and paste it. Sudo apt install Grace. It will install the software. This software will be needed during the Gromacs tutorial. It will show you the graphs while we are working, which means from the Ubuntu you can visualize that how your results are going, how the graphs are showing. Now we will start our tutorial. This is the first page and you need to go to the start of the tutorial. The first step is the prepare the topology. For preparation of the topology, they have provided the file over here. If you click on this, it will provide you a file KALP15 pins. Download this file and paste this file in a folder. I have prepared a folder with the name GMX on the desktop, and I have copied this file over here. The next step we are going to do, we are giving the path to the Linux, that our folder, our working folder, our current working directory is this one. For this, you need to click on this address bar and copy the complete code. After copying, you need to modify it. Like, this is my code. For this, it is the C, I will make it to the small c, remove the colon, and replace all the backward slashes with the forward slashes, like this one. Do with all. You will do it according to your own path. Then we need to add cd space forward slash and mnt. Mnt is for mounting. So our command is ready. When we copy this command and go to the Ubuntu terminal and paste this command over here, it is saying cd go to the drive, this drive, mnt mount, where c in under the c the user, under the user I have HIBSA user, then the desktop, and the desktop there is a folder with name GMX, and press enter. So you can see we are now in this folder. To check either we are in the right side, type ls. When you type ls, it will give you the all files present in the folder. You can see we have placed a single file KALP15 underscore prints.pdb. So we are in the correct folder right now. See, you can check it. This is the GMX folder and this is our file. So now we need to inspect this file. I use the pymol. In the pymol, I have opened this file. Well, in the display, you can go to the sequence mode. Originally you will see the molecule like this one, but you can change the display to sequence mode as residue names, so you can get the names. Or you can check here. If you cannot see this line here, you can click on the sequence and you will get the sequence, and you can rotate it. This means this is a helical protein, this is the NH2 molecule, and this is the acetyl group. This is depending upon the protein. What is your protein? Your protein may also contain NH2 group at one end and carboxyl group at other end. So the protein is perfectly fine. If you find any water molecule, you need to remove these water molecules, because water molecule will not process. It will produce problem at the later stages. Our protein is okay. Now we will move to the next step. In the next step, we need to run this command. We have arranged this command in this way. Copy this command. After copying this command, go to the Ubuntu terminal and paste it and press enter. It will ask you which force field you want to use. If you are using the budget parameter, then it is recommended that you should use at number 13 Gromacs 53A6. But if you are using charm GUI parameters, then it is recommended to use this Gromacs 96 54A7. This is the latest one. For this version, as we are using the budget parameter, we will type 13 and press enter. Now it will ask what is your residue. Amine group NH2. In the previous window, we have told you that they have acetyl group and NH2, which means none of the group is present. So I am going to type 2. But if you have NH3 group, you will type the 0. Or if you have in the second stage, if you have carboxyl group, you will type 0. Like here, it is saying our start terminal is AC1. So you need to choose none. For none, type 2 and press enter. Here, identify residue is NH2. So NH2 is not present, so we choose none. If your residue is present, one of them, you will type 0 or 1. But here we have the different residue, so we are choosing the none. If you have also the different and terminal, choose accordingly. Type 2 and press enter. Please note that when the command runs successfully, Gromacs gives you a quotation. If a quotation appears, this means everything is fine. Here our first step is completed. Now we move to the second step. The next step is topology. Here we need these three files: DPPC 128.pdb, DPPC.ipt and lipid.ipt. As these files are outdated, the Gromacs tutorial has removed these files from here. But you can get these files from our video tutorial link. We have provided all these files. We have placed these files in the folder in which we are working. This is DPPC IPT, this is DPPC 128.pdb and this is lipid.ipt. While these files are produced after the first command running. Now here is one thing. We need to generate the topology. For this we used a force field. Here in our case we are using the force field 53A6, which we chose before at position number 13. But this force field will generate the topology for the protein, which is a single molecule. Now we have another molecule which is known as the lipid, which is DPPC. So we also need to generate the topology for lipid. So we have two things. We need force field to generate topology for the protein, and we need force field to generate the topology for the lipid. As these two are different, so we need two different force fields. So to solve this problem, we generate a copy of force field. This command will generate the copy of force field. If you look into this folder, you cannot find any force field folder. Now we copy this command, this one. What this command do? CP is for copy, and this is our path, and this is our folder, and we are going to generate a new force field. FF means force field. This is the original force field, and we are going to create a copy for lipid force field. Simply copy this command, go to Ubuntu terminal, paste the command and press enter. Now come back to our folder, and you can see now this folder is appeared. This folder we have just created, this is a force field which will be used to generate the topology of the lipid. It contains all the files. So this step is completed. Now come back to our tutorial. We have completed this command. Now there are some changes which we need to do in the lipid force field tutorial. We will explain these changes one by one. Come back to the folder in which you are working, and you have just generated the lipid force field. Open this folder. In this folder, find the file which is ff force field non-bounded.itp. Open it with any text editor you like. I prefer to use Notepad++. This is a short notepad tutorial which makes your things easy. Now you can open it with your favorite text editor. You can use the notepad. I will use here Notepad++. The good thing about Notepad++ is you can see the line number over here, and you can simply search anything you like. Let me close these files. So this is the complete topology file for the protein. Now in this protein file we need to add the atoms from the lipid profile. For this, find out here the lipid.itp. Sorry, it is in the back folder which we just added. This is lipid.itp. Open this file, and this is the topology of the lipid. This is FF non-bounded is the topology of the protein. We need to mix them in a way that it become a single file and Gromacs can read it together. In the lipid topology, these are the atom types, and in the FF non-bounded, these are also atom types. we need to first merge them. In the FF non-bounded ITP, go to the last of this session, like here it is ended at the line 58. After this line 58, add the lines of lipid atom types. Skip the folder, type from here line 14 and up till this. Copy this part, Ctrl C, and place the cursor at line 59 and paste it. You can see these lines are orange. Place an enter to make it separate. Now we have mixed the atom types of protein as well as this part is for the lipid copy. Press Ctrl S. These parts are saved. Now at the second step, we have copied the atom types. Now we need to copy the non-bound form. From this line 29, scroll down until 106, before this line. Again, we need to copy from the line 29 until this line up to 106. Copy this line. What this part was, this is non-bounded parameters. Go to FF non-bounded IPT. This is non-bounded parameters, and look where it ends. It ends, in my case, normally it ends at 1500 something. Yes, here it is ending at 1504. After this 1504, I need to paste the copied file here. You can see the lines are in orange line, and press enter. It will end at 1582. It will start from 1505 and it will end at 1582. Press Ctrl S. The line will be green. It means these topologies are also saved now. Now at next level, the next level is pair type. Scroll down and see if there any other part is present in your protein or in your lipid. I am scrolling down to check is everything else need to be done or this is the last step. In my case, it seems there is nothing more. It is ending at 3070, which means these are the pair types. I need to go to the pair types. We copied till 106, and before this part, we don't need this part to be included in our topology. We need to go to search for the pair types, which is our next step. This is lipid space interaction. This is also needed. Hence, this is pair types, which we need. From this line 676, from this till the end. Also include dihedral type because there is nothing more present in our protein. Our protein last step for pair types. There were no dihedral types, but in lipid we have the dihedral types. So copy all this part and paste it in at the line 3071, including the dihedral angle. You can see these lines are copied 3071 and it is ending 3178. Press Ctrl S to save it. Now the next step is press Ctrl F and type capital H, capital W and find. These are the line with hydrogen water. We need to remove all these line completely. Type and press enter and remove the space. Please note that there should be no space present over here, and then go again and type all the lines which contain hydrogen water because it will generate problem in our topology. Do it one by one by replacing all the hydrogen water. This is we doing the combined topology FF non-bounded topology file, which contains the both topology, protein as well as the lipid. Why we are doing it? We are doing it because we want the combined form of lipid as well as the protein. And we are now removing all the topologies which contain the hydrogen atoms. As you can see here, we are these lines remove. We will remove all them collectively and remove the space, and it is done. You can see it should end 3164. The last line is 3164. The changes are complete. You can now close it. I close the lipid file, I close the non-bonded file, and so on. For your easiness, we have made some point that copy these lines and it will start after 58 line, and then from copy the non-bounded part, it will the line lipid line file are 29 to 106, and it will start from 1505 and so on. We have summarized all these for your tutorial. This will be provided in the video tutorial description link. Now we go to the folder, GMX folder. It has generated the topol.top file. It was generated in the previous step. Open this file, and in this file we need to search this line, include Gromacs, this line. Copy this line, go to file, press Ctrl F, paste this line and press enter. This is the line. This is find at line number 21. Then replace this line with this line. What this line mean? It is for the force field only for protein, but we also included this lipid GFF, which means now it will include the both force field. So we need to give it to the information. Here is the line 21, and replace this with the new line. You can see when you copy and paste, it becomes orange. When you will press Ctrl S, it will become green, which means the changes have been incorporated. Now in the same topology.top file, we need to add some lines about the DPPC.ITP, which means it can utilize the topology of lipid from DPPC file. For this, you need to search this line, copy this line from here, go to topol.top, press Ctrl F, Ctrl V to paste, and find next, press enter. This is the line. These are the line at line number 898 and 899. After this line, add all these highlighted lines, which says cpc.itp. Here is also the cpc.it. We need to remove these two lines and paste the copied file over here. It includes the DPC chain topology and include DPPC IT, and for water topology we have given it the lipid FF, force field and SPC. Press Ctrl S and the topology file is saved. Save this file and control this. With this, we have completed this step, modify the topology file. The topology file, all files have been modified. Now we will move to the next step, which is building the unit cell. Now we are at this stage. Now we need to generate two files. The one file is .mdp file. Click on here. You will find this file. This is minimum.mdp. Copy this name. Go to your folder. Make a new text file. Change the name along with the extension and give it the name minimum.mdp and click yes. If you cannot see the extension, go to these three lines, go to folder options. In options you can see the view. Over here there is an option which is a check mark hide extension. You need to uncheck it and click apply. Then you will see the extension. Be conscious that you need to change the extension. Double click and open it. Now this is copy all the content from here and paste it in here. Ctrl S, save and close. the one file is saved. In the same way, you need to save another file which is with the name topol.dpc.top, which is topol.dpc.top. Copy the name. In the same way, go to the folder, create a new text file, change the name along with the extension, save the name, yes, open the file, copy all of the contents, go to this one and save. After saving it, close this one. Now you have saved the file in the folder. You need to run this command. If you need run this command, you will get some error. We will modify this command with the new one to remove the warning. So copy this command from this file, go to Ubuntu terminal and paste it and press enter, and the job is completed. What this command will do, it will use the Gromacs and will use minimum MDPI file, and it will utilize DPPC 128 PDB file, and it will utilize top.dtp file, and it will generate TPR file which will be used in the next step. In the next step, we will use the trajectory version, and this dpp.tpr file will use in the next step, and DPC 128 will be used and will generate the compact version of this file. Copy this command. This is the next command. Paste it in the Ubuntu terminal and press enter. You will be asked to choose the group. We will choose is the 0, type 0, which is for the system, and press enter, and the job is completed. It has generated a file with the name dppc128_whole.gro. Go to your folder. You can see this is the file. Open this file with a text editor. After opening it, go to the last line, here in the last line you see some values. These are basically the coordinates x, y and z. Go to this file, and these are the basically coordinates. The next command will use these coordinates. The generated coordinates should be placed over at the end of this command. This will use edit configuration, and it will use the KALP processed grow which was produced earlier, and it will produce the new box under the grow, and the new box will be these axes which was generated. So whatever the axes you have generated, the values you generated, you need to add these in your last line of your command line. Add this in your command line. Copy this command line, go to the Ubuntu terminal, paste it and press enter. The quotation showed up, which means the job is successfully completed. With this command we have completed the part up till this. The next part is pack the lipid around the protein. Now we need to generate the file inflate grow. The script is present over here. This is the script. Go to your folder and create a new text file. Change the name along with the extension and name it as inflategrow.py. Save this file, open this and copy all the content from this which was generated from this position. Copy all the content and paste in inflategrow.py, and then press Ctrl S and save and close these files. The files are closed. Now we need to run this command. We have copied this command. It will use the new box grow and it will generate the system.grow. Copy this command, go to Ubuntu terminal, paste the command and press enter, and it is done. Now come to your folder. In your folder we have generated a system.grow. Open this file with the notepad. In the notepad we need to change this number about the total values present in it, but before it we need to remove the line from here, line 141 to 143. These lines need to be removed, and also remove the space, and then go to the last line of this file. Here, this last line. So the total number is 17506, but ignore this one line, 17506. Go to the top, ignore this line and ignore this line. So it was 17506. These are the total line, but we will add 3 lines less because these 2 lines are ignored and the last line is also ignored. So the total number of lines becomes 17503. Press Ctrl S and save this file and close it. So what we did, we have opened the system.grow. We removed the line 142, 141, 142 and 143. We removed 3 lines, and then we changed the number to, total number was 17506 and we have changed it to the 17503. Now go to the tuple.top file again and find this line. Go to your folder and search for the tuple.top. Open this in your editor. Press Ctrl F. Find this line. This is the line. Change this line as this one, which means we are giving a strong position for the inflate grow, and it will produce the strong prose file with include DPPC chain topology and include DPPC IPT. Include DPPC chain topology and include DPPC IPT lines are already present. So what we need to do, we need to add these 4 lines after and if. In this line, after and if, press enter and paste this line. These are the strong position and include DPP chain topology. These are the strong position and these are the DPP chain topology. So our topology.top has been modified, and press Ctrl S and close it. So these changes have been incorporated. After this, we need to generate the restraint file. For this we will use the generate structure. We will use the KALP new box and we will produce the strong pose file with these parameters. Copy this command, go to Ubuntu terminal, paste this command and press enter. It will ask you to choose the parameter file. Here we will choose group number 1, which is the protein. Type 1 and press enter. The job is completed. Now next we are going to use the inflategrow.py system file to generate the DPPC4 system, and it will generate system inflated.grow file under area file. So we will copy this command, go to the Ubuntu terminal and paste it and press enter. The job is completed. Now if you scroll down above, you can see Here there are 128 total lipids, but here it is saying that there are 2 lipids within the cutoff range, which means one will be removed from the upper leaflet and one will be removed from the lower leaflet, which means 2 lipid molecules will be removed. So we need to make changes in the topology file. Go to your folder, working folder, open topol.top file in your editor and go to the last line. In the last line, add this line DPPC 126. Total were 128, but 2 will be deleted, so we will add 126. After adding this line, press Ctrl S and close this file. Now run this command to generate the system with the new information we have just provided. Copy this command, paste in the Ubuntu terminal and press enter. Okay, it's come with the error. Okay, we got it. It says that this file is missing minimum.inflategrow.mdpi. It's short, we just forget it. Here this is the minimization file. Open this file. This is the minimization file. This is minimum.mdp, but we need to change the name according to our command. Copy this name, go to your folder and make a new text document. Change along with the extension, name it as minimum_inflategrow.mdpi and save it. Open this file. It is empty. Now this mdpi file, open this one, copy all the content from here. It doesn't matter if you change the name, just it will use the parameter present in it. Save this file and close it. Now after this, copy this command again, go to the Ubuntu terminal and paste it and press enter. Now it is completed. The error was because we forget to add the parameter file. All these parameter files will be available for you to download. The link is provided in the description, video description section. Now the parameters are fixed. We need to run mdrun. Copy this command, paste it in the Ubuntu terminal and press enter. It is running the MD simulation. It will take few minutes to complete and it is done. It came up with the result that potential energy is -9.5, maximum force is this and norm of the force is this one. Now we run this system inflated grow on the temperature parameter. For this, copy this command, go to Ubuntu terminal, paste the command and press enter. Select 0 for the system, type 0, press enter. After this, run the simulation. The parameters are complete. Copy this command, go to Ubuntu terminal, paste the command and press enter. Now we have placed our protein in the lipid bilayer, but the lipid is too spread out, so we need the shrinking. The next step is shrinking. If you look at this stage, we will shrink it step by step like this one. For this, they have used this command to shrink at the level first, and then they have provided a Python script. For this one, you can run this Python script if you are using the Linux operating system. But if you are using the WSS system, this Python script will not work. For this, we have generated the command to perform it manually from level 1 up to the level 26. For this, we have generated a command. This is the round 1. This complete script file will be provided for you. What you need to do, you need to copy this command. You can also collectively copy them collectively. Go to the Ubuntu and paste it. After pasting it, press enter and it will run the shrinking step, and it is done. The level 1 is completed. In the same way, you will run the round 2. For this round 2, go to the file and it is done. Then the round 3, we have changed the name, that this one is the shrink 1, this one is shrink 2, and this one is shrink 3, and so on. All the rounds are present. All the 26 rounds are present in here. You just need to copy paste one by one. I have run all the 25 steps, and now I am running the step number 26, and this is going to complete. It is completed. You can see the protection energy is now minus 1.1. Initially it was minus 9 something, which means our lipid and protein structure has been minimized sufficiently. All these 26 round of commands are here. You must need to perform each step every time. We have shrinked it up to level 26. Now the next level is to solvate with the water. For solvation, we will use the solvate command of Gromacs. It will copy the system shrink 26 EM grow. As we have shrinked it to the level 26, so we will use the 26. If you have shrinked it to the 20, you will use the command 20, or so on. It will use the topol.top and it will solvate the system. Copy this command, go to Ubuntu terminal, paste it and press enter, and it is completed. Now from here we use another script. Open this script. This is the script. Here is the name water detector.py. Copy this name, go to your working folder GMX, create a new file, text file, change the name along with the text, paste it and press enter. Change the name and open the file. Once the file is open, copy all the content and paste it in your water_deleter.py. Save it and close this file. Once this file is generated, you need to run this command. It will use the water_deleter.py and it will use the system solvate.grow, and it will produce the solvent fix with the water. Copy this command, go to Ubuntu terminal, paste it and press enter. Now you can see that it has removed some water. It is giving information that 1768 water has been deleted. If you are using the different protein or lipid molecule, then this number may vary, and 2188 water molecule remain, which means 4188 water molecule are remaining, and it is giving you the instruction that update your topology. So we will need to update our topology recording the number of water molecules. Come to your working directory and open the topol.top file. In the topol.top, come to the last line. Here it is press enter and make it to the last line, and the water molecule, these are basically solvent or water molecule. These are basically, double check them, these are basically 4188. Add these molecules and save it. Once the file is saved, the water molecule topology has been updated. Now our this step is completed. Solvate with water is completed. The next step is adding the ions. Now we are going to add the ions. For adding the ions, we need to save another file with the name ions.mdpi. Copy this name, go to your folder and create a new text document. In the new text document, along with the extension, give it the name ions.mdpi. Double click and open it. Now open this script, copy all the content from here, go to the ions.mdpi, Ctrl S, save the file and close it. After closing it, run this command. If you run this command, it will give you some error. It is better to use the following command which we have provided because it ignored the warnings. Copy this command, go to Ubuntu terminal, paste the command and press enter. The job is completed. At the next step, we are going to neutralize the charged molecule. For this, copy this command, go to Ubuntu terminal and paste it. Press enter. It is asking to choose the solvent molecules for the group, and it will try to add sodium and chloride ion. For the solvent molecule here, in my case, the group number 15 is solvent. I will type 15 and press enter. It will add the solvent to the, it has added the molecule, like these 4 molecules has been added. Chloride has been added on these following molecules. Now if we come to our folder, this system solvate.ions or grow, I just want to add a little thing. If you right click and sort by the date modified, you will see the most last one generated file over here. If you click on it and open in PyMode, you can see the structure over here. These are the basically solvate ions. If you rotate it, you can see that this is the membrane structure. If I make it smaller, you can see this is the membrane like structure. You can look into it, and these are basically solvate molecule. You can see over here, click on the sequence and go to display and sequence mode residue names. You can find out these are your molecule, and these are basically DPC molecule over here. And if you go on forward, these are basically solvated ions which have been added in your system. So you can have a look how it is going on using this tool. Okay, now our protein has been solvated, which means we have completed this step. This step has been completed. Now we will move toward the next step, which is the energy minimization step. For this energy minimization step, we need to run this command. We will prefer to use this command to ignore the warning. It will use the minimum.mdpi. This file is already present. We have generated a long ago. It is already in our system. It will use the topology file and solvate system, and it will generate the topology. Copy this command and paste it in the Ubuntu terminal and press enter. The topology is the saved, parameters are saved. Now we are going to run the MD run. Copy this MD run for the energy minimization. Copy the command, go to Ubuntu terminal, paste it and press enter. Now the energy minimization has been started. It, depending upon your computer speed, it will take few minutes. Our energy minimization step is completed. This is the potential energy generated. Now this step, energy minimization step, is completed. Now we will move toward the equilibration stage. This is our equilibration step. For this we also generated. If you want to see, there is something additional we have added. If you want to see the potential energy in the form of graph, you can run this command. Copy this command, go to Ubuntu terminal and press enter. Now what parameter you want to see? You want to see the potential energy, pressure, or any other parameter, angle and so on. Here I will type 13 and press enter, and there is no other option I want to choose. Our job is completed. It has generated us the potential energy. Now the next step is, if we want to generate the potential energy values, copy this command, go to terminal and paste the command and press enter. It is asking you which parameter you want to study. Here I am interested in the potential energy. You can also see the pressure, you can see the G96 angle or so on. I will type 13 space 0. It means my only parameter is 13. If you are going to combine two parameters like 13 and 14, you can type 13 space 14. So I am only interested in potential energy, so I have typed 13 space 0 and press enter. The value is generated. If you go to the file, you can see the potential.xvg has been generated. Double click and open this file. You can see this is the potential energy file. Now there is another command, XMGBSA. If you remember, in the start we have installed another program known as XMGBSA. Now it's time to use it. The name of the file is potential.xvg. XMGBSA is the name of the file. Type it. Simply I will copy this command, go to Ubuntu and paste it and press enter. When I will press enter, the graph will pop up. It will give you this is giving me the 400 nanoseconds potential energy graph. You can go to file, print setup and save the file if you want. You can save the graph if you like. Otherwise you can make your own graph with all your parameter using the Excel. I will explain how you can make graphs in Excel to export the data in Excel at the end of this tutorial. This step is completed. This information like XMGBSA and other parameters are not present over here. We have just added it for the better visual effects. Now our energy minimization step is completed. The next step is the equilibration state. Click on the equilibration state. Again, for this we have simplified the command. Copy this command, go to Ubuntu terminal and press enter. It is asking that which parameter you want to use. Here type 1 bar 30, and 1 is the protein and 13 is our DPPC, which is our lipid, which means we are using protein and lipid. Press enter and press Q. Q is quit, and that's it. Our first step is completed. The next step is we are going to run NVT. NVT is basically equilibrium at normal volume and temperature. For this we need to generate a MDP file, nvt.mdp. Copy this name, go to your folder, create a new text document, change the name, open in the text editor. When the file is open, here is the file. Open the file, copy all the content from here, copy and paste it, and Control S save it. Here is the one thing, like you can see this is the nano step. By default it is set at 50,000 nanostep. 50,000 into 2, it's become the 100 nanosecond. It is default. If you want to increase or decrease, just increase the number over here. like if you want to change, you can change reduce 0 1 1 0 from here, and it will automatically reduce the 1 0 from here. So I'm not going to change anything. I'm going to run it by default. Our script is saved. When the script is saved, the next step is running the equilibrium. You can use this command, but this command will come up with the warnings. So we have modified the command for this one, running the this is for the NVT, for to ignore the warnings. Copy this command and paste it and press enter. The parameters are saved. Now we are going to run the NVT equilibrium at NVT. Again, we have modified this command to see the real-time effect. So copy this command. We have added -p. -p gives you the real time that when this command is going to finish. Copy this command, paste it and press enter. Our 100 picosecond step, energy minimization step, is now started. Soon it will give the time. Depending upon the computer speed, the time may vary. You can see that it is giving the time that it will be finished. Approximately it is giving about 15 to 20 minutes it will take to finish this step. Equilibrium at NVT is completed. Now the next step is running the equilibrium part 2, which is at NPT. NPT is normal pressure and temperature. To running this, the procedure is same, but the command is different. For this we need to save this part is completed. Now we would move to the second phase. From the second phase is basically equilibrium at NPT, normal pressure and temperature. For this we again need to save a transcript. Copy this npt.mdp, go to your project working folder, create a new text document. Along with the extension, change the name as npt.mdpi, change the name, open the document, go to the file. Here, this is our script. Open it and copy all the content from here and paste it into the new npt.mdp file. Again, these are the steps. Basically, these are the 1 nanosecond steps. You can, if you want to reduce it to the 100 picosecond, just reduce the one digit lower. I am going to run it as it for the tutorial. Save the file and close it. Also save this file. Now we are going to run this command for NPT. Again, we have modified it to ignore the warning. If you run this command, you may directly from this you may occur some error and your command may not execute properly. So I will recommend to copy this command from here, paste into the Ubuntu terminal and press enter. The parameters are saved. After saving the parameter, the next command is this one. Again, we have modified this command as we have added -p to see the real time progress. Copy this command, go to the Ubuntu terminal, paste the command and press enter. It is taking a lot of time. I think we have done a mistake. Let me check. Oh yes, we have run the next command first. So no worries, if it done, you can terminate this command. For this, we press Ctrl C. This will kill the ongoing command. What we need to do, we are not going to run the md command, but we are going to use this command. Previously by mistake we run the md production command for 1 nanosecond. Please make it sure it is npt.mdpi. So copy this command for npt, go to Ubuntu terminal, paste it. Yes, this is now the right command and press enter. The parameters are saved. After saving the parameter, I am going to copy this command and paste it over here and press enter. The command is started. It will take 10 to 15 minutes to complete. Depending upon your computer speed, it may take longer. NPT equilibration second phase equilibration is completed. Now we have completed the second part, and now we are moving toward the final production state. To produce the final production state, first we need to produce or save the md.mdpi. Copy this name, go to your working folder, create a new text document, change the name along with the extension, name it as md.mdp, open the file. After opening the file, go here, open the link and copy all the content from this and save into your md.mdp file. Same here, this line number 4 represents 1 nanosecond settings. If you want to increase or decrease, you can use it. Normally, for the publication purpose, it is recommended to use at least 50 on 100 nanoseconds. For demo purpose, I am using the same 1 nanosecond. Save the file and then close it. After closing it, now we are going to use this command. This is the production 8th stage. We have modified this command to ignore the warnings. Copy this command. This is the parameters file. Paste it and run it. All the parameters for MD simulations are saved now. Now we are going to use the final run. Again, we have modified this command as -v to see the time. Copy this command and paste it and press enter. The job is started. Depending upon your computer speed, it may take about 1 nanosecond. It will take about 2 to 3 hours or more if you have reduced number of CPU and old CPU. If you have better CPU, then it may complete earlier. Our job is completed. Now we have completed the MD simulation step. Our MD simulation of membrane and protein has been completed. The next step is the analysis. For the analysis, we have different level. First of all, we can determine the deuterium order parameter for acyl chain, density of membrane environment, area per lipid head group, bilayer thickness, and lateral diffusion of lipids. We will see these analyses one by one. First of all, our target is deuterium order parameter, to determine the deuterium order parameter. For this, we need to make an index file. We are going to make 2 index files, to which we are going to name as SN1, and the second index file which we are going to make is SN2. In SN1 and SN2, we are going to choose some groups. In SN1 chain, we are going to choose a selective range of the group. Like here, we are going to choose carbon number 34 to carbon number 50, and we are going to delete carbon from 0 to 21. While in group 2, we are going to choose from carbon number 1 to carbon 31 and delete the atom from 0 to 1. So let's see how we can perform that. First of all, to make the index group, copy this command and go to your Ubuntu terminal, paste it and press enter. It will ask you to select the chain or carbon atom. Here we are going to give this information. Copy all these collectively and paste it into your Ubuntu terminal. It will pop up this way and you say paste anywhere. It says that it has removed these following groups, and over here it has choose the following group. This is the position which we are going to make the SN1 index. It is not going to remove from the original system, but when we will add the SN1 index in the SN1 index, these carbon chains are added and these groups have been removed. Now we have done this. Type Q and press enter. We have successfully generated index SN1. Now we are going to generate a second index chain, which SN2. Copy this command, go to Ubuntu terminal, paste the command and press enter. Now again, the second group, we are going to choose these carbons to be added and these carbon to be deleted. Copy this command and collectively paste it and press paste anywhere. Now in the SN2, it has added these carbon and it has removed these carbon. Type Q and press enter. So now we have generated two index group. One is SN1 and the second one is SN2. We have generated this one SN1 with these parameters and SN2 with these parameters. Now we are going to perform the deuterium order parameter analysis. For first, we are going to run this command. This is for SN1. It will generate a XVG file. Copy this command, paste it, and it is running the analysis and it is completed. When it is completed, now copy this command, go to this one and paste the command and press enter. This is going to use the index2 file and it will generate this file sn2.xvg, and it is generating the file and command is completed. Now the one is deuterium sn1xvg, one file is generated. The second is deuterium sn2xvg. If you go to your folder, you can find this file. This is the deuterium sn2xvg and deuterium sn-xvg. These two files have been generated. If you want to open and see this file, it will give you the information. This is the one file and this one is the second file. If you want to see the plot both files together, you can use the XM grace. Type XM, copy the name from here and paste, give the space and paste it here. Then copy the command from here, the second, give the space and paste here. Then collectively copy this command, go to Ubuntu and paste it. Press enter. It will pop up with the graph. This is the deuterium order parameter graph from the atom. If you look into the picture, it seems like that. It may differ slightly, but this difference is due to the number of steps run in the tutorial. You can add the label or modify it. Now close it. This deuterium step has been completed successfully. Now the second step is the density of membrane. If we want to measure the density of the membrane, how we can calculate the density? For this, we are going to make one more index file with the name density group. Copy this command, paste in the Ubuntu terminal and press enter. It is going to ask which groups you want to include into the index file for the density of the membrane. For this, we are going to copy this. Here we are saying that we are using the 13 and carbon 1, 2, 3 and C4. These are the information we are going to paste. Copy this command and paste it here and press enter. Now we need to name it. We will name it as a head groups 20. Paste it, it will give it the name 20. The second group we are going to create is the acyl group. For the acyl group, we are using the group 13 and we are including these carbon step by step. Copy this command, paste it and press enter. For this one, we are giving it to the name 21 glycerol ester. I have given it to the name group name 21 and group as glycerol ester. Now the question is why I'm here I gave the name as 20, here I gave the name as 21. Why I choose this number? I choose it from the index 5. You can see here at 19 numbers are already filled with these information. The very next number is 20. So I gave it the 20 number. The next number I have gave is the 21 number. And in both the 13 in the name 20 head groups and as well as 21 head groups, I choose as a 13. Then why I 13? What is 13? 13 is basically my lipid. I am targeting my lipid, which means I am using the lipid, and from lipid I am using these atoms. Here I am also targeting the lipid. I use the 13 and these carbons are used. At the third level I am again using 20 and 21 these groups, and I am going to name it as 22 as acyl chain. So I have created 3 groups, group 20 with head groups, group 21 with glycerol ester and group 22 with acyl chain. You can also make more groups as 23, 24, 25 and so on. When you are done, type Q and press enter. Our job is completed. So we have generated these groups as per the head groups, the acyl group and these groups. Now we are going to run it to make it center. Here they have given us simple example of dense head group at XVG, but it is not complete. For the above group, we have generated a separate command each time. What you need to do, you need to run these commands one by one. This is the head group, this is the dense glycerol group, and then this is the acyl groups, and then we have made it a center, again centralized and so on. What you need to do, you need to run it one by one. Copy this command and complete. It will ask you to choose the select the group to center density profile around. For this we will choose 0 as a system, type 0 and press enter. Again type 0 and plus enter. It will calculate. So run all this command one by one. I am going to demonstrate that how you are going to use it quickly. Then the next command, after this one, then this one, then this one. We have completed all the, I think we have completed all the 6 commands. Then we are going to look into that. You can see you have generated the groups into your folder: density group, dense head group, dense glycerol group, then acyl group, dense water and dense protein. All these groups have been generated. Now I want to see the plot for each of these one. What you need to do, you need to copy this name, this name, this name and any other name. Like here I have used densyl head groups, dense glycerol group, dense acyl group, NXY, dense water and dense protein. Dense water and protein have been already generated during the first command we run in this folder. When we run this first command, the water and protein groups are already present. I just copy these names and added these two protein in my name, density water and density protein. Now I am going to copy the complete command and paste it in the Ubuntu terminal and press enter. When you press enter, you can see a line. The line is not clearly visible due to the different coloring parameter. I think we have made a small mistake over here. Let me do it again. Well, over here, this is the first density head group XVG. Copy this command and paste it again. When it asks for the group, this is the density head groups. So basically we will look into the head groups. So instead of choosing the system, we are not going to choose the system, but we are going to define it for 20 for better visualization. If you go to the system, it will collectively give the complete system, but here we are only interested in the 20 in the head group. So type here 20 and again 20. For the next one, this one is for density glycerol group. Copy this command, paste it, and this is glycerol ester. For glycerol ester it is 21. Here it is, type 21, enter, again 21, enter, and it is completed. The next group is density acyl. Copy this command, type it, and here is the acyl chain as the 22 group. 22 and then again 22. The next command is for the water. This one is for the water. Copy over here, look into the water. Water is at the position number 15. So type 15 and again 15. And the last group is for the protein. This is the protein. Type here the protein, look into the protein. Protein is at number 1 position. Type 1 and then type 1. The job is completed. The groups are updated. Over here, see, the old groups has been labeled as the hashtag and the new groups are labeled as here. All these groups are updated. Now we are going to again copy these dense protein XVG, water XVG, acyl XVG, glycerol XVG, and dense head group XVG. Use the XM graph and copy this graph as -xy to arrange it all together. This command will collectively give a single graph for all the values. Now paste this command and press enter. Oh, sorry, I have run the same command for protein again, so I need to cancel it. Control C, it will make the command cancel. Copy this command and paste it and press enter. Now you can look into that. It is giving the different graph, the blue bar, the green bar, the red bar, and the black bar. It is just like the same, it is appearing in that. The rest of information is their editing. You can make graph edit from here or file over here. Now this analysis is completed. The next analysis is area per lipid and bilayer thickness. It is currently unavailable. It is a separate tutorial. we will cover it later on. The next thing is later diffusion of lipid. The diffusion of lipid molecule, we will cover it. This graph need a grid mat MD if you want to look into it. It is a complete new script for GitHub. We are not going to cover it today. In the future video, we will make a separate video on this one. But today is our last thing is later diffusion of lipids. For this one, copy this command, go to Ubuntu terminal and paste it. It is going to make a new index file with the P8. From this one, we are going to choose ASP8. Paste it, P8. 128 atom found with name P8. Press Q and press enter. The job is completed. Now we are going to run the MSD calculation. This command will tell us that how molecules of lipid move across membrane and the protein. So I am going to copy this command and paste it. Type the Control D and it is ended. Oh sorry, I have typed Control C which ended the command. So we are going to run the command again. This was our command. Control C, copy the command, Control V, paste. Here we are going to type 20 plus enter and type Control D. When you type Control D, it will calculate and give you the values. The job is completed. If you can see here, it is RMSD underscore PDB. Simply we can copy this name along with the extension. Type a new command to see the plot. If you have, we can use the command of xmgrace space msdp8. Control C, go to the Ubuntu and Control V, paste it and press enter. So this is the value which we are going to expect from it. This is the graph of XMD value. This is the tau protein and picosecond. This is the MSD value and this is the average mean. So from here, this tutorial is completed. This is the summary. You can read it if you like. Close this graph. The one thing I want to tell you that these are the values. If you open it, you can also see the values from here. All the XVG files are basically your results. You can open this file in the Excel sheet. Let me demonstrate you. Make a new Excel sheet. Open this Excel sheet. Once the Excel sheet is open, go to the file, open, browse. In the browse, I have a folder GMX. In the GMX, type here to the all files. In here you have the file with the name XVG. Look for all the XVG files. Search here XVG. You can see these are the XVG file. Let's suppose this is MSDP8 XVG file. select this and open it. When you open it, this window will pop up. Click on delimited, then click next, click on the space, then click next, and then click finish. Now you can see all the values are pasted in a separate menu. You can select these values, all these values are selected. You can go to insert and then go to scatter chart and make a chart you like. You can add ligand and you can add more information you like. So this is our tutorial about the membrane protein using KALP15 in DPPC. If you have any query, please ask in the comment section. If you have not yet subscribed our channel, please like and subscribe. We will also upload other Gromacs tutorial as well. Stay tuned for the more recent and advanced in silico modeling tools tutorials on our channel. Thank you so much for watching. Allah Hafiz.